Okay, another biology lab report that I'm going to mark now. As always, red is all the bad things, green are the good things. I would like to thank the student for giving me permission to mark this on YouTube, and those post-its are simply there to cover up the name of the student. So let's get started, and I think I'm going to do it a little bit faster because the other videos are quite long. So the research question right on the front page: to what extent, English problem, okay, do the different colors of light emitting diodes affect the number of oxygen bubbles? So basically this student changed the color of light and then counted the rate of photosynthesis. So the number of oxygen bubbles is not relevant here, it's the rate of photosynthesis. Okay, and the light emitting diodes, I mean, that's part of the method, and also the oxygen bubbles, that's also part of the method. So yeah, we don't care about the oxygen bubbles, we care about the rate of photosynthesis. Significance and topic and research question is what I read is pretty good, and I'm just going to show you some of these things here. She's using the water plant Elodea, also known as Egeria densa. Wrong, mistake. This is always in lowercase, okay? So Elodea is the name of the water plant, and she justified this quite well, okay? Basically how she got the interest and is to find out other light sources. So basically, can you not also use other light sources to optimize photosynthesis? And she wanted to check this, and I think this is a very, how shall I say, yeah, that's a very consistent, and I think, yeah, I like this, it makes sense. So the next thing is the following. The background information here talks about the importance of wavelength. So there is a connection to the research question here, however. Okay, chlorophylls which are optimized for different light colors, it's also fine, but what is missing here is the clear link to the research question to oxygen production. Does it mean that if you use the optimum light, then you expect the highest oxygen production because oxygen reflects the rate of photosynthesis? This connection is not made, and that's not so good. Okay, so there is the last link is missing. I mean, it's all relevant if you know what the experiment is about, but there is no, yeah, you have to have this knowledge in advance, otherwise you don't really know. Why is she telling me all of that? Okay, so that is, yeah. Oh, down here, a light is used in the process called photolysis when water is split to create oxygen, hydrogen ion. Is this the oxygen that you want to use, that you want to measure? Is this the oxygen that you want to measure? Yes. But you have to say that, okay? There is no connection here. So this can be actually done by simply writing an additional sentence. So then down here, yeah, safety and ethical issues. Now look at this. She wrote me a whole paragraph. In reality, there are not many safety or ethical issues. Still, she tried to justify this. Okay, she said that what she's going to do with the LOD after with the water plant after she did the experiment, yeah, I do not consider it very convincing, at least where we live, is a waste of water and so on. But you know, she showed an attempt to actually also address safety and ethical issues, and this should be awarded. I think that's good. So, overview of the method, good. Before you jump into any details, you have to give the reader an overview of what the experiment is even about. Okay, I intend to count the number of oxygen bubbles, okay, depending on the LED, okay, and then here again the problem with the name, and you have to italicize this, okay. So this is 5 centimeter long pieces, that's a control of variables, control of variables, control of variables, and the LED is also controlled by yeah, using the LED screen, okay. Control of variables, control of variables. My suggestion would be, if you want to get a little bit more points, and I hope she's doing this later down here, okay, is a Y10, Y25, and so on. So just justify this, okay? So that is something that I'll be looking out for. Justify the values. Okay, or are these just arbitrary? I know these are arbitrary values sometimes, but then just say that, okay? Why 5 centimeters? Why not longer? Why not shorter? Okay, I'm going to be looking for these things. Materials used, okay, she's writing a paragraph here. I normally write a bullet pointed list, but that doesn't matter. I need iPads because iPads, you don't, yeah, are made of LEDs. Yeah, okay, English issue. They're not only made of LEDs. Other than the light source, I intend to use a ruler. Okay, why? Okay, to measure. Here she justifies it. Okay, she justifies it. It's a timer. Okay, Yes, and so on. Okay, I think yeah, she is justifying the material, so I think that's also okay. I mean, I don't know, maybe you could kind of combine this a little bit, but it's okay. Yeah? Control of the controlled variables. So all of those variables that have to stay the same, okay? Which you need to keep the same. All of those introductory sentences you can just cancel out, of course. You need to go, it's the distance, okay? Of course, you already told me that the distance has to be controlled. How do you control this? How? How? Okay? She justifies it here. That's good. But how do you actually control the distance? Prevent this from happening, I use the ruler to make sure the distance in the test tip is always 10 centimeters. Okay, here she does the justification. Okay, I think that's okay. Yeah? She uses a ruler to do that. Another factor is the plant's the same length. Okay, so here she justifies this. I think that's okay. Okay, I'm not going into details now. I'm just going to skim through this a little bit. The independent variable, that is the different light color. Okay, the color of the LED, that's fine. How do you change this? Okay, how? We have 3 trials. Okay, that's the number of trials. No, yes, but okay, yeah, okay, fine. But how do you actually change? How do you actually make the iPads actually light up in different colors? This is really, there's something missing here. I don't understand. Okay, so this is and the number of trials is not an independent variable actually. I mean, okay, the trials, that's fine to have this here, but actually it doesn't belong in here. Okay, this is, I don't know, somewhere else, yeah, but not in here. So the dependent variables are the oxygen bubbles. Okay. I will be counting how many oxygen bubbles are produced every minute. Each trial will last for 5 minutes. Why? It's fine, but why? Why 5 minutes? Justify. Okay. Can prevent having more errors. It's time here she justifies it. Okay. It is time. Okay. See? I'm already taking this again. Yeah. So at least I'm looking for some kind of justification here, but yeah, I don't know how does she actually put the water plants into the water? There are still many things missing, so I'm not quite happy with this because maybe a drawing. Yeah, she talked, I think, about, what did she say here? In the method, the materials used, the length, the timer. Don't you need water? Don't you need some kind of a container where you actually put the plant in? How does she keep the plant down? I don't know. Okay, this is very unclear. Okay, so raw data. Okay, so let's have a look here. This is the raw data table, and the first thing that I already see, why does she not combine this? The format is not very good, I think. Okay, it's very confusing, the format. I think it's possible to combine this a little bit. The numbers could be centered. I mean, it's kind of difficult to actually, excuse me, see the difference between time and the actual bubbles here. Okay, I mean, these are just numbers, so I don't know. There is some kind of a layout formatting issue. Okay, and the other thing is the following. Here you have the average per minute, and then all of a sudden here you have more significant digits than you actually have counted here. So the average cannot have more significant digits. Here. This is all nothing. Okay. So here what I need is not so good. This is not good. Okay, that's not good. This has to look better. Optically, it has to look better. It just confuses me. Why trials here? Doesn't it make more sense maybe to have the trials on top and then the averages on the bottom? I don't know, but I don't know. I don't like this so much. Same issue here. It's kind of weird. Why does this go on the next page? This has to go on the previous page. Although, yeah. Okay, it can be seen that the LED is the most, really, how can I see this? There is no reference to the data. You got to tell me the values, where? Okay. So, oh, come on, look at this. This is the way on how to calculate the average. It's the same issue. People really like to convert. It is irrelevant. You can assume that people know how to calculate an average. It's not a math paper. Average number, red, green, blue. Why? I thought you already told me that. I don't understand. And here again, inconsistent number of significant figures. This is not good. It should be centered. The optics really have to be improved. What about the title? The caption? Okay. It is proven. I don't like if students use proven. I'm a TOK teacher as well, so I don't like proven. Okay. It could be demonstrated. Okay. Look at this. The printout is a disaster. I cannot read this. here as well. Okay, the printout should be better. Measurement uncertainties. Excuse me again. The biggest measurement error is the count. Why? What's the problem here? There were not that many bubbles per minute. So why did you miscount or what? In a dark room with only the timer and the colored LED as a light source. Okay, so it's actually the human error that you're. How big is this problem? How big? How big of a problem? I mean, this is not very. Yeah, so that is a measurement uncertainty, but I was actually kind of hoping that you would talk about the standard deviation here. It's not only a measurement uncertainty from the counting perspective, but actually also from the oxygen. So maybe some of them are larger, maybe some of them are smaller, and larger ones maybe you have to wait longer. There are many more issues that you can talk about. I don't like this so much here. Graph, of course, a graph, but give it a meaningful title, not just graph. Okay, here is the graph, and I already see a major error of understanding, and this is such a common thing. Okay, look, the color down here, the color is a so-called, it's a non-continuous variable or discrete variable. That means you have categories, okay? So this is supposed to be blue, red, yellow, and green. You cannot even see the labels down here, okay? So that's optics. But the thing is when you have a discontinuous variable, then you cannot, first of all, draw a line like this. It does not make sense. You can only do this if you have a continuous variable. How can you convert this into a continuous variable? By actually not having separate colors down here, but wavelength. But if you have wavelength, then basically the green should be somewhere in between blue and red. So the sequence here is actually doesn't. Let's say that I chose to put the green somewhere else. Then you don't have this line anymore. So this is already a major problem that the student even attempted to draw a line here. Okay, that's simply because she tried to do, or he tried to do that. When I told the student draw a line of best fit, actually doesn't make sense here. The student did it anyway. Okay, just in order to fulfill some kind of arbitrary criteria, which actually doesn't make sense here. Okay, so this is completely out. This line is totally wrong. The next thing is the following. A second big problem are the standard deviations here. Okay, do you actually see these are the standard deviations, the error bars? They are all the same size because the student used Excel, and actually this standard deviation bar is the standard deviation, which is the same in all cases, but is basically calculated by Excel by calculating the standard deviations of all of these averages. But that's not what I want. So the Excel kind of averaged all of those averages. We already have the average, but Excel averaged them again and then calculated from that the standard deviation. This doesn't make sense. What we want to have is if you want to make these error bars, then you have to do the following. You have to calculate a separate standard deviation for each item. And then the error bars will be different. And so you're not allowed to click on the standard deviation checkbox in Excel because this is going to do something different. You got to almost manually set these error bars. Okay. That's a common problem, big problem, okay, because it doesn't make sense, okay. Look, how many trials does she have? She only has 3 trials anyway. I doubt that it makes a lot of sense even to calculate a standard deviation from these, okay. Standard deviations only make sense, I think, from if you have about 7 to 10 different repeats. It's not very meaningful here. You know what? For the sake of our purpose, it doesn't matter. Okay. So my advice here would be not only have the averages, but also have a standard deviation column. Okay. And for each trial, no. Time in minutes, average per minute. Heaven's sake. Heaven's sake, I'm just seeing this. No, this is completely wrong. Sorry, guys. Oh my gosh. Look what she's done. This is a cumulative... look, it's like this. In the first minutes, there were 3 bubbles. In the second minute, she continued counting. I think. Because the values always go up. In the second minute, including the first minute, there were 4 bubbles. So there was actually only 1 bubble per minute here. And then you have basically an increase in bubbles going up, and then you cannot calculate an average because this is a changing trend. I'm just seeing this myself right now. This is a fundamental problem in understanding. Okay, you cannot calculate. No, this does not make sense. Oh my gosh, oh my gosh, oh my gosh, oh my gosh, this is totally out. You look, the point is the following. If you have a trend, okay, over time, yeah, something going up, okay, like this, then you cannot calculate an average over this. Okay, that's not possible because we're changing the time. Yeah. So what the student could do is the following. I mean, that would be possible. She should do the data processing in such a way that essentially she collects it as a raw data and then calculates back. In the first minute, she has got 3 bubbles. In the second minute, it's 4 minus 3, which is 1. And in the third minute, it's 7 minus 1 minus 3, which is, there were 3 bubbles here, right? No, 7. Oh gosh, I'm getting confused now. You got to calculate this back. How many bubbles were actually made in the individual minutes here? Okay, and then you can calculate an average. But then you calculate basically, you don't even have 3 trials, and you have essentially you have 12 trials. Okay. Okay, this is completely out. So forget about this. So this graph doesn't even make sense. Okay, this graph doesn't even make sense in this case. Because you cannot calculate an average over a changing trend. No, not this bothers me. Okay, so look at this. You've got in the first minute 2, in the second minute 3. This means that actually in the second minute she did not count 3, but in the second minute, including the first minute, there were 3. This means that there must have been 1 bubble here. Okay, this is 5 bubbles. So this means there is 2 plus 1 is 3. There must be 2 bubbles here. Okay. That we had here, and this is 6, so this is 3 plus 2 is 3 plus 2 is 5, and there must be 1 bubble to get 6 bubbles. Okay, and because there was no change, there's no change here, 0. Okay, so it's 2 1 2 1 0, and then you can calculate an average of this, and these are basically in the first trial run the average number of bubbles per minute. Okay, I think, yeah. Because this is so-called cumulative data, okay, and you cannot simply calculate an average over cumulative data. It's not going to work. Wow, okay, this is a major thingy. So let's move on. Quantitative data, the leaves turn darker, okay, than before starting in relation to the different colors. Has been were hotter, okay. Were some colors hotter than others? In case because blue, because blue is closer to violet color and violet has the shortest wave, thus resulting in high energy. Actually, yeah, okay. Actually, water warms up on the opposite wavelength, infrared, and it's not ultraviolet, but maybe that could be as well. Low energy, but actually the red wavelength actually heats up water a lot because it's closest to infrared. But no, it's depends on the individual frequencies. Okay, but at least there was some kind of an attempt here, and also an attempt to link the observations to the how do you say to the colors. Conclusion. Oh, okay. I'm spending more time on this. You know what I'm going to do is I'm just going to cut this short. Just looking at the whole thing, this is way too short, okay? The strengths are probably not evaluated, it's simply listed here, but evaluation means that you have to indicate how much of a problem was this, so I want to have more here. Don't forget, you get a lot of points here, and you cannot simply, I don't know, this is less than a page, you cannot simply expect a lot of points, almost a third of the points simply by writing 2 paragraphs. This seems to be some kind of theoretical background. Okay, this, I don't know. Yeah, yeah, I don't know. I don't know to what extent this is still valid considering the data. Okay, so generally I want to have more here and more concrete concrete improvements. Okay, yeah, I'm just going to cut this a little bit short because I already see that there is bibliography. Wow, a lot of stuff here. But the problem that I have with that is if you actually have all these things, then you also have to cite and reference them, and it has not been cited and referenced a lot. Well, actually I promised myself to actually do this only in I don't know, about 7 or 8 minutes, and now I'm again almost 20 minutes marking the whole thing. Yeah, but in any case, my verdict. I have to give another verdict again. Yeah, I mean, there's some parts that are pretty good actually, yeah, but actually then some parts the whole data thing completely collapses. You know what? Medium, room for improvement. That's my verdict. Even though I have to say that probably some parts are quite okay. So I actually have to give smiley faces and frowny faces, yeah, on the different sections here. But overall, this needs some improvement, and I think there is a lot of possibility for improvement. And this lab report really has potential, because also the personal engagement is pretty good, because it's actually an experiment that the student actually was quite involved with herself. Okay, so that's it. Wish you a nice day. All the best. Okay, bye bye.